17, and that is citric acid cycle. Okay. Last week we did study about the glycolysis. Okay. Glycolysis. That's on the glucose catabolism. Catabolism. I don't know whether I can get this one clear for you onto your document camera. See in the document camera what I have written. Could you see clearly or not? I'll change my pen. Perhaps that will help. The first part is the glycolysis and the glucose catabolism. That means the glucose is breaking down, and it ends in glycolysis. That's on the end of glycolysis. Where we stopped at the end of glycolysis, we stopped with pyruvate. Do you remember? Pyruvate. Pyruvate, okay. Now, we are going to start from this pyruvate. That means we end of glycolysis and we are going to start from pyruvate into another pathway. From pyruvate. So this is our starting point now. From this pyruvate we are going to study further catabolism. As I mentioned before, we are studying the metabolism. Metabolism comprises of anabolism and catabolism. And then here we are going to study about the catabolism of pyruvate. Okay. How we are going to study the catabolism of pyruvate? We are going to introduce the next step is the TCA cycle. TCA means here T stands for tricarboxylic acid. Very TCA. That's A here. TCA cycle. Because the cycle means, as a cycle, it comes out one compound, another compound, another compound, and then another compound, and then another compound. And finally, you are synthesizing the same thing. So it's a cyclic event which is taking place here. So the compound 1, 2, 3, 4, and 5, and then from the 5 again you are going to synthesize the compound 1. So this process is going on and on and on and on, and that will produce more of different energized compound that we are going to see today. And the starting point here is pyruvate. Okay. So as I mentioned, the metabolism means you have got all the enzymes. So that we need to study about what is the TCA. I put in short TCA cycle or Krebs, K-R-E-B, Krebs cycle. See, it's a scientist name, Krebs cycle. Probably you might have studied in the earlier class in biochemistry, Krebs cycle. And this cycle, this TCA cycle or citric acid cycle, is the final common pathway. You should another term, final common pathway. So you should always remember what is the final common pathway. You should remember that TCA cycle is the final common pathway. For what? Pathway for oxidation of fuel. Pathway for oxidation of fuel. Okay. Now, what is the fuel in an energetic or in a nutrient fuels? What generally we refer to is carbohydrates. Carbohydrates, and proteins are amino acids, and fats are fatty acids. I put FA, fatty acids. Okay. So these are the nutrients what we eat, and they are ending in where? Final common pathway for what? For oxidation. So the TCA cycle plays a major role in intermediary metabolism. That you should understand. And most of these nutrients, the end product of carbohydrates, or amino acids, or fats, or fatty acids, okay, they enter the gateway or the enter point. Enter at gateway. Means a compound called acetyl coenzyme A. I'll write it in a separate paper. Acetyl coenzyme A. Okay, acetyl coenzyme A. So this is the entry point of carbohydrates, end of glycolysis, the pyruvate to next step is acetyl coenzyme A, and fatty acids. This is the entry point at the end of the catabolism of fatty acids, and then at the end of the catabolism, some of them are amino acids, also acetyl coenzyme A as the entry point or the gateway entrance to the metabolism. So we are talking about the TCA cycle. The first and initial steps in our first compound will produce in the acetyl coenzyme A. Okay, that's what I want to make sure you follow that. Because we are going to step by step, and this is a very interesting mechanism today we are going to discuss. That's in the steps of pyruvate dehydrogenase complex enzyme. Okay. In your textbook, if you just see the the subcellular organelle which is involved here, subcellular organelle, which is mitochondria, because the TCA cycle is present inside the mitochondria. Okay, TCA cycle is operating. The cycle is operating inside mitochondria. So now I want to recollect your histology or cell biology class. What is the structure of mitochondria? Okay. See, mitochondria, it is made up of, as I said, if you see like this as the outer membrane, and then you can have like an inner membrane like this, like cristae. Okay, this is the outer membrane, and this is the inner membrane of mitochondria. So the enzymes mainly present in this inner membrane of mitochondria like this. Otherwise we call it as a matrix. So here also, the matrix enzymes are present over here, and they also in the membrane level, in the outer membrane. So the TCA cycle membranes, they are present in the matrix. This is matrix. Okay, so they are present in this matrix of inside the inner membrane of mitochondria. So the outer membrane and the inner membrane plays a major role forming the ionic gradient. What ionic gradient? It can transport the proton inside out or outside in, and thereby they can maintain the gradient. And when the proton is pumping out, you have the ATP synthesis is going on, or ATPase activity. This part we will study next class, that is on chapter 18, on the detailed mechanism. But right now, you please remember that TCA cycle enzymes are present in the matrix. And now we are going to study onto the TCA cycle. Okay? So the site of operating this cycle inside the mitochondria. We will go into the detail of your textbook, the PowerPoint. I will just get one thing here. So this is again a cycle. TCA cycle looks like that, but this is a roundabout in a city. So this is don't ask me that is it present inside the mitochondria or not, but just yes, a picture, an art picture of that. Okay. Now we are going on to this acetyl-CoA, which is entering onto the roundabout. And I don't know whether how many of you are familiar with the roundabout. Here we got mostly on signals, red signal, green, and where you can follow through. In England, they don't have, in most other places they have roundabout. So as soon as you enter, you have to There you have to follow left side. So you have to go on the left side round and round, round, round. You have to go like this. Suppose if you are entering from here, and then you have to turn left side, right? So you drive like this, and then if you have your exit, you can go here, and then another exit, you can go here, another exit, you can go here. So like that. So if you miss one exit, man, you have to go again round and then get into that exit. So it's a round and there are 3 or 4 lanes. So yeah, you should be very careful selecting your lane. So if you go outer lane, that go here. Middle lane, you can go here. And then inner lane, you can go here like that. So it's a roundabout, but it's the easy way to do work on that. But sometimes if you get confused, people going round and round, round and again, they will come back and then get into that. So that's on the roundabout. And then here in this one TCA cycle, acetyl coenzyme A is the entry point over here. And we are going to study the intermediate one by one. And what happens here, the net part of the cycle is 2 molecules of carbon dioxide is released. And then 8 electrons is being released. And GTP is there. And then the cycle is going on and on. But every time the cycle going on, one acetyl-CoA molecule is getting inside into the cycle. And then is a carbon dioxide release. And then it will go and then form another another molecule of acetyl-CoA. So this acetyl-CoA is the entry point for carbohydrate as well as the amino acids and fatty acids. Okay, that's the schematic representations of this picture. This is acetyl coenzyme A structure. You can see that one acetyl coenzyme structure. This is CH3CO acetyl, and this is the entire thing is the coenzyme A. See, this is a sulfur atom which is coming up. Okay. This is the acetyl coenzyme A molecule over here. This is the entire thing which is coming up. Now, yeah, this is the mitochondria. That's what I want to show you here. You can see the one which is the middle. These are the cristae, or the inner membrane of mitochondria. The outer membrane is the smooth surface. The inner membranes, you have got the ridges. Okay, it goes like this, it comes like this. So it's a folded structure, meaning the inner membrane surface will be large, but it is just like folded inside inwardly. And inside this, you have more of a TCA cycle enzyme or tricarboxylic acid cycle enzyme. This is the real electron micrograph of mitochondria. Okay, now the next one, what we are going to study here. Overview of the citric acid cycle. Overview. What happened in the mitochondria? Okay. As I mentioned before, 2 carbon units. 2 carbon units. They are oxidized. That's why I explained earlier the carbon dioxide is released. 2 molecules of carbon dioxide is released from 2 carbon. So that means 2 CO2. So 2 C and then oxygen is there. So it's the 2 carbon is oxidized to carbon dioxide. And it also produces 1 molecule of ATP, and you have high energy electrons. High energy electrons. Okay. That form high energy electrons, they form NADH and FADH2. So NADH and FADH2, they are high energy electron, and 1 molecule of ATP is formed. When you see the overview of the TCA cycle, it is not producing much of ATP, not producing more ATP, the cycle itself. However, it produces what? High energy electron, high energy electrons, and these electrons now will be used. More of these electrons are produced by the cycle, and it will be used in the electron transport chain, ETC, electron transport chain, inside the mitochondria, and there it will, the electrons are more used for ATP synthesis. But as such, the TCA cycle not of, except the ETC, electron transport chain, up to this point, we don't see any ATP synthesis, direct ATP synthesis, except one ATP in this cycle, okay? That you should remember. So don't think that TCA cycle produces more of ATP. That is not correct as a direct way of interpretation. But however, it produces high energy electrons. That helps to produce more ATP, not directly. Okay. So in other words, TCA cycle, harvest. Harvest means it will produce more, harvest high energy electrons. That's what electrons, high energy electrons. So these high energy electrons are used to produce ATP. So TCA cycle is more of high energy electrons in the form of NADH and FADH2. So this is very important. This is the net result of TCA cycle. So now we will go into a little bit of the TCA cycle. The TCA cycle, again, is the central metabolic hub, which I already mentioned earlier. It's the central metabolic hub, meaning this is a meeting point. It's the meeting point of different nutrients and the catabolic product, and everything you should come through this TCA cycle. But amino acids or fatty acids or carbohydrates, anything. But this is the central. So the studies of intermediate metabolism, whatever you are studying in the entire semester, and this is the central role. We are going to see in a different occasion for different metabolism, we are going to link every time TCA cycle, TCA cycle, TCA cycle, and one way or other we are going to link it. So that's what I want to give you much importance to clear out the doubt now. Okay? Now, the cycle is important. Why it is important? The cycle is an important source of building blocks. That's very important. In the life science, building blocks. Different compounds are produced, building block of several amino acids. Several amino acids, the origin is coming from TCA cycle. Several amino acids. And nucleotides, the DNA producing components, nucleotides, origin from TCA cycle. And porphyrin, that is also there. Porphyrins, which is very important for heme biosynthesis, for hemoglobin, heme biosynthesis. Heme biosynthesis. This is for the RBC and the hemoglobin, for oxygen transport and everything. So the origin of porphyrin again is coming from TCA cycle. Okay. The another product, oxaloacetate. The oxaloacetate, another compound, is produced by TCA cycle, and this is a precursor for glucose. Precursor for glucose. Have you studied the gluconeogenesis? In this step, the oxaloacetate compound is very important. So if you eat much of fatty acid or fat or oil stuff or even proteins, those amino acids and fatty acids, they can produce the water for oxaloacetate, and from these oxaloacetate precursors, and glucose will be produced from that. So even if you're not eating carbohydrates, your body can synthesize the glucose molecule for survival. So that's another point we will study in later on in a different perspective of the glucose synthesis. So glucose production as well as the porphyrins, and then also amino acids, and all of them, TCA cycle plays a major role as a central hub. Okay, and also TCA cycle produce what? Oxidation, the result of oxidation of carbon dioxide, carbon to carbon dioxide, and also it also produces the high energy electrons, and that is used for ATP synthesis in the mitochondria, ATP synthesis. So the importance of this particular TCA cycle: produces energy, produces high energy electrons for ATP, produces carbohydrates. It also helps to getting into the amino acids or the protein synthesis pathway, as well as the DNA, nucleic acid synthesis pathway. All of them depending upon the TCA cycle. So if you study the TCA cycle, means you have done a wonderful job on intermediary metabolism. Okay. So as I mentioned before, it is neither generate large amount of ATP or nor includes oxygen as a reactant. So TCA cycle is not producing ATP directly, or it is not using oxygen directly. Okay. But this cycle, however, what this cycle will do? This cycle removes electrons from what is the initial product which we studied, acetyl coenzyme A. Otherwise we call it acetyl-CoA, CoA. This is the A, acetyl-CoA, ACETYL, acetyl coenzyme A, and uses the electron. It removes the electrons. Mind it, the TCA cycle removes the electron from acetyl-CoA, okay, and uses these electrons to form NADH and FADH2. So now we have to see where the NADH is coming from, in which step we have FADH2 is being produced, and the electrons are being transferred. Okay. So in the steps of transfer of the electrons, it produces 3 hydride ions. That is, what is hydride? 6 electrons transferred. 6 electrons transferred to 3 molecules of NAD+. Meaning, if the electrons are coming here, then the products will be 3 NADH and then H+, which will produce it. 3 NAD+ will produce NADH. I will write it separately, NADH. So that is the product. Okay. NAD+. Here the electrons is coming joined together, and then you have a NADH+ and H+. And also one pair of hydrogen atom that will join with the electrons, with the 2 electrons. We have 2 hydrogens, so 2 electrons together join to form into the FADH2. So FAD plus H2 is the electrons, and then for FADH2. So in that by it will produce more of reducing equivalent. We call it as NADH+ or FADH2. These are the molecules or electron carriers. These electron carriers will transport in the mitochondrial distribution and the ATPase enzyme. We will study how the electron transport chain is working with these electron carriers of NADH and FADH2 to produce ATP next class. Okay, but the source at that time is this NADH. Just like what we studied last week, we studied the glucose is metabolized and catabolized into pyruvate, and pyruvate is the starting point for today's class for the TCA cycle, and then next class the source for the electron carrier electron transfer chain is the FADH as the net result of TCA cycle. So that's why I am connecting one metabolism into the another metabolism, another class. So these electron carriers, how many electron carriers which we studied here? Here 3 NADH and one molecule of FADH2, and they will yield in the mitochondria how much? 9 molecules of ATP, which we will study there. 9 molecules of ATP. So that means we are getting more energy when they are oxidized by oxygen, in presence of oxygen. So this oxygen is coming from you, whatever your respiration from the air, that oxygen will help to produce of more energy of ATP, electron transport chain. And when there is an oxygen, that we call it as cellular aerobic respiration, cellular respiration. When the cell is taking oxygen, that means that uses the NADH and FADH2 in the mitochondria, produces of this amount of ATP. So 3 NADH will produce of 1 FADH to produce 9 molecules of ATP, and these NADH FADH2 is coming from TCA cycle. So TCA cycle will feed. Where the TCA cycle is coming from? That is coming from glycolytic pathway, glycolysis. So it's one to the other, which is being linked to each other. Okay. Now the link for all this, the link. What is the link here? Glycolysis, and here the TCA cycle. So there is a link compound which we call it as the glycolysis. The end product is pyruvate, right? Which I mentioned, pyruvate. And the pyruvate is being activated by an enzyme that is called pyruvate dehydrogenase enzyme. I'm writing it here. Pyruvate dehydrogenase, GENASE, it's a long name. Pyruvate dehydrogenase enzyme. Otherwise, they call it as a PDH, pyruvate dehydrogenase enzyme. And we are going to study about this pyruvate dehydrogenase enzyme in a minute. Okay. Do you have any questions so far? You follow? Victoria? Syncorange? Up to this point. Now I'm going to the first step of TCA cycle that's on pyruvate, how the pyruvate is converted to acetyl-CoA by the pyruvate dehydrogenase enzyme complex. I'm going to discuss now with you. Okay. If there is no question here in Sugarland, no question? You follow? No? You're not following? No? Okay. Now, the first step, pyruvate. Pyruvate plus coenzyme A, coenzyme A, plus 1 molecule of NAD plus. Okay. So these are the reactants, all of them reacting together. And the product here, the product for this reaction is acetyl coenzyme A. See, coenzyme A, it is attached to the acetyl coenzyme A, okay, plus carbon dioxide is being released outside. That's what the respiration, whatever you do, the carbon dioxide coming out of your exhaled breath, that's coming carbon dioxide, plus NADH and H plus. These are the product now. So now you please concentrate on this. So the pyruvate, where the pyruvate is coming from? Coming from end of glucose catabolism or end of glycolysis. And the coenzyme A is there in the mitochondria inside the matrix. And inside the matrix, NAD plus also there. So the enzyme which is present over here is the pyruvate dehydrogenase. Pyruvate dehydrogenase enzyme, PDH. This enzyme is a complex enzyme. Complex. Okay? Meaning it will normally one enzyme will do one job with the reactants, okay? But this enzyme complex will do, there are 3 small enzymes together, put together to form a complex. But all of them will do one function, pyruvate conversion to acetyl-CoA in 3 different steps. It will do it, okay? So those things are very interesting, like a swinging arm hypothesis or swinging arm the method of transfer. So I'm going to get the PDH complex, which is from the E. coli. They have studied extensively, they have studied pyruvate dehydrogenase enzyme in E. coli. And the electron micrographs also you can see on the figure 17.5. Okay, we'll see. I'll show you in a bit here in the PowerPoint. Back to the PowerPoint. Mitochondria inside the matrix. Now, if you go the next one. Now, this is the matrix, inner membrane and outer membrane. This is the structure of mitochondria. Could you see the inner membrane of mitochondria, which is being folded like a cristae? They call it cristae. Now, let's go into how the C2 means here. This is acetyl coenzyme A, which you have 2 carbon unit, and this is the oxaloacetate. It's a 4 carbon units. So 4 plus 2, how much? 6. So now you have a 6 carbon units here. And then you have a NADH is released, this step, and carbon dioxide is released, and C5 and NADH is released, another molecule of carbon dioxide is coming out, and C4, GTP is released, and then FADH2, and then that then convert to NADH. And finally the C4 is again produced. That means oxaloacetate is continuously produced by the cycle. And then another molecule of C2 will be taken from the outside. So like this, the carbon dioxide, whatever the C2 acetyl-CoA which is coming, enter into the cycle, eliminate carbon dioxide, 2 molecules of carbon entry and 2 molecules of carbon dioxide is released, but the 4 carbon oxaloacetate is regenerated. So that's why it is called a cycle. So if I ask you to write an essay of the cycle, then you have to mention why we call it cycle and what is the regenerator, what is the carbon which is being converted into carbon dioxide, and how many molecules of NADH is produced, how many molecules of ATP is produced, and how many molecules of FADH2 is being produced. All of them you can write it in maybe a 1 short paragraph. Okay, that's why that in a nutshell we can study on the TCA cycle. So each entry point, this C6 and C5, C here and then C4, different entry point you have amino acid may enter, or this is the acetyl-CoA with the carbohydrates can enter. So the different entry point we will study later on. Okay, that's why the central role on this. And now you can see citric acid cycle and oxidative phosphorylation. This part we are going to study next week, but today we are going to concentrate on the TCA cycle alone. And this one is the inner mitochondrial membrane. You can see that matrix and then inner mitochondrial membrane. So the enzymes are present in the matrix side. Fatty acid, acetyl-CoA. Glucose, acetyl-CoA. Amino acid, acetyl-CoA. So all of them converted to acetyl-CoA. Now we are going to study what and how the acetyl-CoA is being produced from pyruvate, and then we are going to study how it is being introduced here on carbon dioxide. Okay, and this part we will study next week. Now we are concentrating on the one part, one time, the TCA cycle. Okay. Okay. Now, pyruvate entry. What is the carbon dioxide removal? We call it this carbon dioxide removal is the decarboxylation. First step in this enzyme mechanism is carboxylation. Decarboxylation. Carboxylation means addition of carbon dioxide, and decarboxylation here is removal of carbon dioxide. Carbon, that's why it's removed. And also the electron is removal means oxidation. So this step, removal of carbon dioxide, is the decarboxylation. The another one is removal of electrons, 2e. Okay, the removal of electrons means, that is called, in other words, it is called removal of electron is called, we call it as oxidation. So if you think the removal of carbon dioxide called decarboxylation and removal of electron called oxidation, when you combine these together, what is they call is, what you can call it, the name of the reaction is oxidative decarboxylation. Oxidative decarboxylation. Which enzyme is having the capacity, or which enzyme will do the function of oxidative decarboxylation? The enzyme is pyruvate decarboxylase. Decarboxylase. L-A-A-S-E means the enzyme pyruvate decarboxylase enzyme. Okay. And it is a large enzyme in the large molecule. It's very large. And it composed of 3 distinct enzymes because it does different function, right? One enzyme, but it has many function. So 3 distinct enzymes are present. 3 distinct enzymes are present over there. Do you follow now? Oh, you can't see now here whatever I'm writing it here? Could you see? No? I'm sorry. Okay, now. Could you see now here? Pyruvate decarboxylase. Victoria, you follow? Pyruvate decarboxylase, large distinct 3 enzymes, yes? Hello? Anyone?
Yes.
Yes, okay, good. Because I'm writing on the overhead projector here, could you see from there in your end? Or you can see from here?
Yes, sir.
Oh, you can see that other end. Oh, okay, good. Because I didn't reduce my screen here, so that's why. So 3 distinct enzymes, okay. Now, the part, I'll go through once again. Pyruvate and then acetyl-CoA to carbon dioxide, GTP, and electrons removal. What is this one? This is the crystalline structure of pyruvate decarboxylase structure. It looks like ribosomes, but it is not ribosomes. Looks like ribosomes. That means the large molecule. If I say large means you can clearly see that is the capacity of pyruvate decarboxylase enzyme structure, crystalline structure. Okay. Now, the pyruvate dehydrogenase complex of E. coli, 3 enzymes which I mentioned earlier, right? This is a pyruvate dehydrogenase component E1, that means enzyme 1. Number of chains is 24. Prosthetic group means the active group present in the enzyme. I want to explain this. Suppose if this is an enzyme, okay, enzyme is a protein molecule, right? It's an enzyme. Among this enzyme, certain region of the enzyme will be highly active. Suppose this is the part of this enzyme will be attached to the prosthetic group. We call it prosthetic group. That's what is a prosthetic group. Here we call it a TPP. Okay. Thiamine pyrophosphate. TPP, in other words, it is called thiamine pyrophosphate. I'll write it here in another one. Thiamine pyrophosphate. Thiamine pyrophosphate means TPP in short. See, thiamine is again it's a vitamin, right? Vitamin B complex, and one of the vitamins B1 is a thiamine. So thiamine pyrophosphate, it is present in the prosthetic group, and that means this is the part where the reaction, or the active site of the enzyme where the reaction is taking place. So this is the number 1. You have to remember that too. Okay. Now the second one is the dihydrolipoyl transacetylase enzyme. Dihydrolipoyl transacetylase enzyme. In other words, we call E2. That is the second enzyme in pyruvate dehydrogenase enzyme complex. Number of chain is 24 again, but the prosthetic group is lipomide. And what this will do, transfer of acetyl group to coenzyme A. What is the function of this enzyme? Active group is oxidative decarboxylation of pyruvate, which just now I explained to you. Oxidation, that means removal of electron, and decarboxylation is removal of carbon dioxide. Both together, oxidative decarboxylation of pyruvate, that's carried out by one enzyme, that enzyme number 1. In the complex, there are 3 enzymes. First enzyme will do this, and then the second enzyme on the prosthetic group is lipomide, and that is acting as a transfer of acetyl coenzyme A, a transfer of acetyl group to coenzyme A. That means it join together. The result of oxidative decarboxylation of pyruvate is acetyl group, and then coenzyme A is addition, that is carried out by lipamide. Because I'm going to ask some questions from this table, which is responsible for the addition of coenzyme A, TPP or lipamide. Your answer is lipamide. So that is the answer. And then the third part is the dihydrolipoyl dehydrogenase, that is enzyme 3, 12 chain, and then here the prosthetic group is FAD, that flavin adenine dinucleotide. That's called flavin adenine dinucleotide. Otherwise we call it FAD. It will do regeneration of oxidized form of lipamide. So this is the lipamide, it will regenerate. First, after the transfer group, lipamide may change, but this FAD will help to regenerate this lipamide formation so that it can reuse for this cycle. So the enzyme reaction, which is a fascinating one of the footprint in biochemistry, means the people extensively studied this particular enzyme. It's very interesting enzyme complex. I love it now. So let us go into the mechanism in a further detail, okay, and you will enjoy that. Okay, what is the thymine pyrophosphate? You can see the thymine molecule and this pyrophosphate, this is the group, and lipoic acid. Imagine these, all of them are present in one enzyme complex, and this the lipoic acid is a long chain. When combined together, it's like an arm. You know, it will pick up in one arm. Suppose if you are existing here on one enzyme, imagine, and this side is another enzyme, and then here is another enzyme. What will this compound will do? Pick up one particular reactant, the product, and put it in the next active site, and then it will remove it, and then you put it in the another one. So what it will do? It will swing the molecule in a direction. It will change from one active site of one enzyme into the another active site of another enzyme. So it basically swings around the protein. We call it a swinging arm mechanism. That's what we call it in lipoic arm, lipoate arm. Right. Pyruvate minus decarboxylation, removal of carbon dioxide. C double bond O, C double bond O, removal of carbon dioxide. Now this is the acetyl group, CH3CO acetyl group. Oxidation, again 2 electrons. Again, acetyl-CoA, here the coenzyme add, transfer of CoA, acetyl-CoA is formed. So this formation of acetyl-CoA is the first step in TCA cycle, and it is interesting. And the next one, carbanion, not carbon ion. Just to check the spelling, carbanion of TPP. Here the carbanion, and then pyruvate combined together to form with the hydrogen plus hydroxyethyl TPP, and that is being transferred to TPP, carbanion, pyruvate, addition of the compound, and this is the steric hindrance, how the electrons are moving from formation of one compound into the other. Some of them are unstable, but the stable form will be one which will react with another compound. Okay. If you see the hydroxyethyl TPP, lipamide, and carbanion TPP, acetyl lipamide. Don't worry about this intermediate. We are worrying about the end product, but just what happened inside the enzyme. These are the mechanism. Okay, don't worry about this part. Coenzyme A, acetyl lipamide, acetyl-CoA, dihydrolipamide. Now look this one. Reactive disulfide bond and lysine side chain. See, lysine side chain is a long one, and reactive disulfide bond here. This is the one reactive. Imagine from here to here is a lengthy one. Just like, that's why I said this long arm, so that the large molecule, you can take one particular compound on the active side into the another. This lengthy chain will help you to move around, and flexible too. Okay, lipamide. And the next one, dihydrolipamide to lipamide. See the SH, this is a sulfur atom. Now is a bond in presence of FAD, FADH2. And then NAD plus, and the FAD is being again synthesized, and NADH is being produced. Okay. This is the structure of pyruvate decarboxylase enzyme complex. E1, E2, E3. Could you see that black one, alpha 2, beta 2, alpha beta, and alpha 3. See, you can see green, yellow, and pink. All of them put together in one enzyme complex. So 3 different enzymes binding together, closing together in a one complex fashion, where the enzyme is acting from one active site of this one enzyme to the another active site of another enzyme, another active site of another enzyme. So 3 different reactions, 3 different enzymes, but they are present in one, which is linking all these 3 things is the lipomide. Okay. Now I'll show the interesting part. Lipomide domain. So where the lipomide is present is the lipomide. So you can see the structure of the lipomide. Now this one, which will go this one A trimer. Okay, from here, and trans-acetylase reaction here. This compound will join and it will transfer from one end to the other. Imagine. So this is the enlarged picture. What is happening inside? So that's what it is. The structure or the crystalline structure of this, of secondary, primary structure of the protein together. It looks like this. Okay, but swinging arm lipomide will be moving around. How? Here, this is the mechanism I'm going to explain to you today. As I mentioned earlier, yellow point, green, and the pink. Okay, 3 enzymes. The color represent 3 color. Yellow, green, pink, 3 enzymes. E1, here the E2 and E3. Okay. First reaction, TPP and FAD, and this is the lipoic acid. Okay, double bond. Sulfur bond. So this one, watch this disulfide. This one, it will go all over the place and produce the same one, lipoic acid. First reaction, decarboxylase, pyruvate decarboxylase reaction, where the removal of carbon dioxide. How? TPP. Here, acetyl-CoA here, okay, joined together on the enzyme 1, and here nothing happens here, okay. And look, the second reaction, this one, which is present outside, which is joined together on the enzyme 2 complex, push it in to where? Enzyme 1. So this is the active side. Now there is a reaction going on, the removal, everything. Could you see the next step, 3? Where this arm which is going up to here, the yellow, and now it is insert the next reaction into the pink again. It's coming to E2. Could you follow now? Here the E1, and then now it's coming to E2. But this E2 of one enzyme here, and this is another trimer, say another one. So it moves. It will take a compound. See, sulfur attached to CH3O, acetyl group is joined to this sulfur. And then now it is going to another subunit, another enzyme, E2, another subunit of E2 enzyme. And then the fourth step is the coenzyme A, okay, is being removed from here. And now you can see the, it goes on the FAD part, SH and SH. Could this see the arm, where the arm will go? Here, FAD and then SH and SH. And then fifth step, what happened? FADH2 is produced, and again this one is regenerated, okay, this step. Could you see now here this one, and then the NAD and the NADH2, the FADH2 will be, here the hydrogen will be removed at the 6th step, where the FAD also produced, and the same one, original one, the first step where we produce the lipoic acid, that is also being produced here. So this is the fascinating step where one lipoic acid from one enzyme unit, and then how that transfer in the different step and then back. If you imagine, if it is there, it will pick up in one arm and put it over there and pick up that and put it with another arm, and then it will regenerate itself. So it goes along with the swinging arm hypothesis, or swinging arm mechanism of this one. So now I would like to explain what's happening one by one, and that is the end part of this mechanism. Okay. So what happens here? 3 enzymes and 5 coenzymes are present. Coenzymes. 3 enzymes and 5 coenzymes. Could you see that one here? Okay. And the mechanism is swinging. You know, have you played swing in your earlier childhood? Swinging mechanism. Swinging arm mechanism there. Otherwise we call it as a flexible linkage. No other enzyme reaction you will experience this part. That is on the flexible linkage. This will allow. Which one is a flexible linkage? Which compound is a flexible linkage? Is lipomide. Lipo, L-I-P-O-A-M-I-D, lipomide reaction. To move, lipomide to move, okay, between active sites, So that's what is happening, moving around one active site into the other, lipomide. So I'm referring to this is called lipomide. So it attached to this one, and then look at that one here, one part, and then it's getting in here, and then getting there, and then FAD, and then regenerate, and this is also regenerate. So it's a very, very interesting point. That's what I said. This is the the part of biochemistry of swinging on hypothesis. And the first part, what happened in the first step, pyruvate decarboxylate. Pyruvate decarboxylation. Pyruvate decarboxylated and carbon dioxide leaves. That's what the first step in the carbon dioxide. What happened in the second one? It happens in the enzyme 2, right? That enzyme 2 inserts the lipomide arm. I put lipomide arm in where? Lipomide arm in E1 site. Lipomide ion in the E1 site. So that's what is happening here. Okay, good. Now the third step. What is third step? E1. Now the enzyme 1 catalyzes transfer. Catalyzes the transfer. E1 catalyzes the transfer of acetyl group. Acetyl group to what? Lipomide. Lipomide reaction. And the acetylated arm leaves. Now the acetylated arm leaves E1 and enters E2 Okay. Okay, now, so look at the E1 catalyzes the transfer of acetyl group to lipomide, and this arm leaves E1 and enters E2. Okay. Now, reaction 4. At the fourth step, what? The acetyl moiety. The moiety means group. M-O-I-E-T-Y. In the biochemistry, we call it acetyl moiety, phosphate moiety, carboxylate moiety. So always we call it moiety, means a part of that particular reaction or the product. Okay, or the chemical group, we call it acetyl moiety, is transferred to coenzyme A, coenzyme A. Now this acetyl moiety transferred to coenzyme A, so result is acetyl-CoA. Now the leaves, acetyl-CoA leaves, where? It leaves the E2 enzyme. Okay. Then the next part is the reduced lipomide. When you remove the reduced lipomide, L-I-P-O-A-M-I-D, lipomide, swings, that's what it is, it does, swings to E3, another step, swings to E3, lipomide, okay. Number 4. And now the next part is number 5, fifth reaction, fifth step. I write it up. At E3, what happened? Lipomide is oxidized by coenzyme FAD. FAD coenzyme is involved oxidized, and this will reactivate it. Result is reactivation of lipamide. And this lipamide is now ready for the another reaction, ready for another transfer. I put it, transfer. So that is the recycling. And the final product, NADH, that happened in the sixth step. Step number 6. The final product, NADH, is produced with reactivation of FADH2 to FAD. So FAD we start, right? And then the FAD is again reactivated. Again, it's been getting at the end product, and the FADH2 to FAD, and also we reactivation of lipamide against for the another transfer. So everything is being now circulated into the cycle of the pyruvate dehydrogenase enzyme complex. So this part, whatever we studied here, now it's from what is called pyruvate to acetyl coenzyme A. So this is the first step, and this is the most important step, because this step is pyruvate to acetyl CoA is irreversible step. Irreversible step. In biochemical pathway, certain step is reversible and is irreversible, and it is control or regulate the TCA cycle because of the irreversible pathway. Regulate the TCA cycle. So that's why I want to explain the mechanism or swinging arm hypothesis with this one here. Okay. So what I'll give you 10 minutes break, and then come back, and then we will start the rest of the TCA cycle. Okay. Do you have any question? Victoria? Everyone? Okay, let us go into the next step. I'm just reviewing what we have done onto these 5 paths. Is the TPP, this enzyme 1. Now we can see the first step, the carbon dioxide removal, and then the acetyl-CoA is attached to TPP, and the next step is this one is getting in. Look at this, is inserted here, and now this reaction is going on. It's going in and picking up this part from TPP, and then swing down into this. Look at that, step 3. See, this is the one enzyme complex, how this will swing from TP1 to here, and there is a reaction going on where the coenzyme A is being added to it, okay, and been released. CoA is added and it is released. So this blue part, acetyl-CoA, is released outside. Now you have SH and SH, so it will go switch to FAD here, and then the hydrogen from here, it will be over here, FADH2. And now this one will come back after that. See, the bond is formed, SH is, hydrogen is donated, and S is here. And then FADH2, again this step, NADH, and H+, and that is going back, and the FAD is again produced. So what happened here in this one enzyme, I'm doing it in a different steps, but everything is taking place in one enzyme. This will go here, and here, and here, and then this is being reduced. So imagine this is the cycle which is going on the pyruvate decarboxylase enzyme cycle. Now, the next step in this pathway, let us work on the citrate synthase. As I mentioned earlier, acetyl coenzyme A is being released, okay, as the previous step which I explained to you. And this acetyl-CoA now, it's going to react. That's the first step, as I mentioned, is being combined with oxaloacetate oxaloacetate. Oxaloacetate, another compound, and this oxaloacetate is a 4-carbon unit. Acetyl-CoA is 2-carbon unit. Okay, so 2 plus 4, you go to 6, and this is being done, or the uniter of 2-carbon to 4-carbon unit. We call it as a synthase reaction. That is called the citrate synthase. Citrate synthase enzyme. This enzyme is participating, uniting these together and to form citrate. So the enzyme that produces citrate, we call it as a citrate, C-I-T-R-A-T-E, citrate. So citrate synthase is an enzyme and citrate is the product. Well, how the citrate is being produced? The acetyl coenzyme A and oxaloacetate together and form the citrate compound. Okay. Citrate, the synthase means, again, I want to explain here. What is the synthase means? Okay, here. What do you mean by synthase? If you want to understand what is synthase means, two units, okay, reacts each other, or binds together, okay, binds together without ATP. That is very important. Normally, any compound, mixture of compound together for the union, you need to put some more energy. But here, in this synthase molecule or synthase enzyme, will act with the acetyl-CoA 2-carbon unit and oxaloacetate 4-carbon unit. It join together without any ATP expenditure. So meaning the synthase is a highly reactive group, citrate synthase. So when acetyl-CoA is produced, the acetyl-CoA plus oxaloacetate, the citrate is being produced now. So now, where are we now? We are on the citrate pathway. Okay. And the conformational changes in citrate synthase are in the oxaloacetate. So what happened here is the citrate synthase produce citrate from oxaloacetate and acetyl-CoA. Now this citrate, the next step, the citrate is now isomerized. Isomerized, meaning, what is meant by an isomer? Molecular formula is the same, but the structural formula is different. So the citrate structure is going to change by isomerase enzyme. That is the term we call it as isomerized. Isomerized by an enzyme. The enzyme for isomerization, we call it as aconitase. A-C-O-N-I-T-A-S-E. Aconitase. Okay, this is an intermediate of the citrate into isocitrate through aconitate. That's why we call it aconitase, the enzyme here. Okay, here I'll write it here. Citrate, I put SIT, just an abbreviation. Citrate into aconitate, and then aconitate to isocitrate. So citrate is produced by citrate synthase, and the citrate now is isomerized to isocitrate. The intermediate, we call it aconitate, and the enzyme which involved here is aconitase. That's it. So now, where are we? We are at the point of isocitrate. Citrate and isomer of citrate by aconitase enzyme. So we call isocitrate. Now, the next step from isocitrate, this isocitrate, now is oxidized. Okay? Now, I'll write it here. Isocitrate, now oxidized. Oxidized and decarboxylated. Oxidative decarboxylation, just like a pyruvate dehydrogenase enzyme complex. Here, oxidized and decarboxylated. Decarboxylated means what? Oxidized means removal of electrons. Electron is removed, and decarboxylation means removal of carbon dioxide. So it will remove, and then it will lead to alpha ketoglutarate, alpha ketoglutaric acid or glutarate. So in short, we call it a alpha Kg, mean not kilogram, but it is a alpha ketoglutaric acid Kg, plus NADH plus carbon dioxide. So these are the reaction, the alpha ketoglutaric acid. And the enzyme which involved here in this reaction, which is isocitrate to alpha ketoglutarate, we call it as alpha ketoglutarate dehydrogenase. So the enzyme which is carry out this reaction, that is called alpha ketoglutarate acid dehydrogenase enzyme. Okay, where you have NADH produced and carbon dioxide is released. That's the part. Now, where are we now? We are in the alpha ketoglutarate acid dehydrogenase, and the product for this one, okay, the product for alpha ketoglutarate acid is going to be changed into succinyl-CoA, succinyl coenzyme A. So this is the product from alpha ketoglutarate acid, alpha ketoglutarate acid, and alpha ketoglutarate acid dehydrogenase, the enzyme, dehydrogenase enzyme. It will produce by a succinyl coenzyme A. And this succinyl coenzyme A, this one is a high energy compound, high energy thioester, okay. And it has the delta G value. Delta G is the free energy value, which I have studied in the first class on this, minus 33.5 kilojoule. That is the amount of energy is being released. That means this succinyl-CoA will produce more of high energy, free energy is being released. That is used for ATP synthesis. Now, succinyl-CoA to succinate, that's the thioester. So, the next step, succinyl-CoA, succinyl coenzyme A plus PI plus ADP will produce, what is the product for this? Succinate. Succinate. See, the coenzyme A, whenever the coenzyme A is there, it's a high energy bond. So that meaning it can produce, it gives the energy to join the inorganic phosphate to ADP to produce succinate and ATP. So here the energy ATP is being produced in here, and plus CoA is released. So this CoA is again recircled. Okay. Now the next step, the succinate. Now the succinate is going to be changed to oxidation. Okay, the pathway. Succinate. Succinate, they have oxidized to fumarate. Fumarate, another intermediate in the cycle, fumarate to malate, and then the malate to oxaloacetic acid. So now the oxaloacetate is being regenerated, okay, and this oxaloacetate now is going to combine with the acetyl-CoA, which is another molecule of new acetyl-CoA is entering into the mitochondria, and then what happened together? It forms citrate, synthase or citrates. Using the citrate synthase enzyme, you get citrate molecule produced, and it will enter into the another cycle on production of the high energy molecules. Okay, and the oxaloacetate regenerated by the oxidation of succinic acid. The succinate by oxidation. By oxidation. Oxidation means again removal of electron. Whenever I say oxidation means you should remember that is on removal of electrons. Okay, so please remember that removal of electrons means here the oxidation. All right, the next one, next step. what happened, the overall idea about the cycle. Regulation of TCA cycle. Now the TCA cycle is the step is over. Now I will review it here in the PowerPoint. Some of the features. This we have studied. Okay. And oxaloacetate, acetyl-CoA, citryl-CoA, and citrate. These are some of the intermediate which is formed, which is not stable, and this is stable where the coenzyme is released. And now this is the structure of oxaloacetate combination, where the oxaloacetate binding into the site, citrate synthase. This is the citrate synthase, and there's two binding sites. Okay, these are the cartoon explaining the enzyme complex. Now the next one, the substrate complex, how this oxaloacetate and acetyl-CoA react into an enzyme in molecule. These are the cartoon where the diagram showing the reaction is taking place inside the active site of the enzyme, citrate synthase, enol intermediate, and how this steric of change of electrons. This arrow means the electrons or bond is shifting to one side, and thereby the compound is released from the enzyme and react with the another compound. Okay. And the enol intermediate, citryl, and then citrate to cis aconitate, and then isocitrate bonding. Okay. So look at this one, CHOH, and then here COH and H. So that's it. Just a change of the position. We call it citrate and isocitrate. Why we have to change this to this? Because this will, next step we are removing the carbon dioxide. If you have the CH means the bond is so tight, it will not release the carbon dioxide. That's why you have to have change of the position of this H and OH from the second carbon to the third carbon, the hydrogen. And the COH and CH means more of steric instability, and it will remove the carbon dioxide in the next step. You can see that next step. See how the citrate, the molecules, how they rearranging these citrate and isocitrate forming into the, see now the isocitrate, NADH, now this part, look at this H and OH, that's being there, and oxalosuccinate, and then another molecule of hydrogen remove the carbon dioxide. Where is the carbon dioxide? The carbon dioxide here. This carbon dioxide is removed here. This is called alpha ketoglutaric acid. Structure of alpha ketoglutaric. This is a keto group, alpha ketoglutaric acid. Now the next step, alpha ketoglutarate plus NAD plus and a coenzyme A, we have succinyl coenzyme A. So COOH again, COO minus, that is being removed. The blue carbon dioxide is removed in this step. So succinyl CoA is being produced, and now the succinyl CoA, the next step is to succinate and the inorganic phosphate and GDP form, and then succinate and CoA and GTP, and from GTP to ATP is formed from ADP. So this is the first step in succinyl CoA oxidation. And the next step, we can see this is the mechanism how the succinyl CoA is being transformed to GDP to form GTP. Okay. This again how the energetic value, alpha beta subunits, succinate fumarate, and succinate from succinate to fumarate. See, you can see the two hydrogen. These two hydrogen is being removed by succinate dehydrogenase enzyme, that is the FAD and FADH2. You can see that from FADH2. And then if you remove this hydrogen, this bond is being used here for a double bond. So fumarate is an unsaturated compound, fumarate. And then fumarate again with water, you get malate, and then again a malate dehydrogenase enzyme, you have hydrogen and NADH. So finally you produce oxaloacetate. And this oxaloacetate now recircle to synthesize citrate compound with acetyl-CoA. New acetyl-CoA is combined to oxaloacetate. So that's why the cycle is finished. So we are regenerating whatever the molecule we used, and the cycle is going on and on and on and on. Okay. For this, he got Nobel Prize for the cycle. And now here this is a mechanism how the fumarate is being converted into malate. This is some of the mechanism of the chemical. Now this is the entire reaction of TCA cycle. Look at the citrate watch the green stuff, CH2COOH, aconitase, CH2COO, and then isocitrate dehydrogenase enzyme, alpha keto dehydrogenase enzyme complex, succinyl-CoA synthetase, succinate dehydrogenase, fumarase, malate dehydrogenase, and citrate synthase again, another molecule of acetyl-CoA. So the cycle is going on and on. So you can see the one NADH, another NADH, ATP or GTP here, FADH2, and another NADH. So all of them, you know, it put together, this is cycle form, and this is present in all over the an aerobic cycle, we call it. And this is the entire citric acid cycle reaction. Please remember these steps. You know, you will get some questions from this particular table. This entire thing, what I explained to you, it is in a table form. That's it. Porosity group, iron sulfur. FeS means iron sulfur. We are going to see the iron sulfur group in the next class as well. Okay, and this is another way of regulation, how the glucose is converted into pyruvate, and pyruvate to acetyl-CoA, and acetyl-CoA to carbon dioxide, and lipid is supplying to acetyl-CoA, and acetyl-CoA to lipid biosynthesis. That means fatty acid, whatever the fatty acids which you are eating the fat, that can be converted to on this particular step. Have you ever heard of Atkins diet, where you can eat enough amount of fat, you don't eat carbohydrates, so you cut down all this part, but you are taking more of lipids or fat that enter here acetyl-CoA, that will produce more of energy, but not from the glucose side. That's what they subtract that, because if you have enough of excess of carbohydrates or glucose or any starch, that will hold more water, and that will give increase your weight, and tissues and everything is become more of soggy. So the Atkins, they said, okay, eat only meat or no carb, then you can reduce the weight. That's okay for a while, but after some time, excess of protein that knock off the kidney, so you will suffer from kidney disease or kidney malfunction later on. So Atkins diet is okay for a temporary, but it is not a permanent one. So again, if you think of any other amino acid that also entering the acetyl-CoA, it will get, you know, it's a temporary situation on that part. But in regular situation, you have to eat everything together. Active PDH, pyruvate dehydrogenase enzyme, is the inactive PDH. So that means the kinases, ATP to ADP, the kinase, if the kinase is more active, you become PDH is inactive. When phosphatase is more active, you have phosphatase reaction to the active PDH. So this is very important in a cancer biology, for the cancer carcinogenesis. You know, if more of this particular phosphatase or you know, phosphate dehydrogenase enzyme, pyruvate dehydrogenase enzyme is actively participating in some other cancer. So they always work with kinase inhibitors and phosphatase inhibitors, and then they can manage the situation of the carcinogenesis. So this is interesting concept, and we'll see how the high energy charge and low energy charge, how the pyruvate is adjusting towards the promotion of ATP formation. Here the high energy charge, you can see that is negatively regulated. Acetyl-CoA will block here, and then if you have more ATP, block here. You know, NADH block this one, and thereby it will maintain the same energy level. Suppose your energy is going down, what will happen? The pyruvate will activate pyruvate dehydrogenase enzyme complex, NAD to NADH, that is coming out, and then acetyl-CoA here, and this electrons being used, and this is the one, the ADP, more of ADP in the cell, that meaning, you know, you don't have much of energy that will promote, and thereby the phosphate, the pyruvate dehydrogenase enzyme is highly activated to turn more ATP synthesis. So that's the regulation of this particular step. Okay. High energy charge, the same one what I explained earlier, the other cartoon. And here pyruvate with excess of ATP, acetyl-CoA, NADH block. Minus sign means block. So block the conversion of pyruvate to acetyl-CoA enzyme A. So if you have enough energy, ATP, more energy, you have energized state, that will block, because there is no need to produce more of acetyl-CoA. Suppose if you don't have this energy, if you used of, you need of more of energy, so what will happen, if you have the ATP will be produced, so less amount of ATP, so it will be forward reaction, it will produce of more and more and more. So ATP and everything is less, this is being promoted. So that's why whenever you do much of exercise, we are using of more of ATP in your body, and thereby promote the acetyl coenzyme A, and this will, depending upon your excess of oxygen, whatever you are breathing it out and in and out, more of oxygen. When you do the exercise, you are using more of ATP, so you are burning the calorie and thereby activating the step, and thereby you are producing the cycle efficiently. At the same time, if you don't do any exercise or excess more of stuff, of food and fatty stuff and everything put together, it ends in acetyl-CoA, accumulation of acetyl-CoA, and it will not get into the oxidation process, but it reverses of acetyl-CoA to form aceto-acetyl-CoA and fatty acid synthesis, and you get more of adipose tissue, become obese, and everything is happening. So the TCA cycle form a major intermediate metabolism here. Okay, acetyl-CoA is very important here. We will study the fatty acid synthesis later on in our class. And here how the oxaloacetate getting into the aspartic acid and purine, pyrimidines and other amino acid synthesis. As I mentioned earlier in the class, how the oxaloacetate is a product is very important for synthesis of glucose as well as synthesis of other amino acids and also DNA components. So pyruvate will also can directly convert to oxaloacetate. Now oxaloacetate citrate and fatty acid sterols can also produce from citric acid. Alpha ketoglutarate, that's entering into the glutamic acid, other amino acids and purine can synthesize. So succinyl-CoA to porphyrins, heme and chlorophyll molecule, that is derived from succinyl-CoA. So if you study TCA cycle, you study the entire biochemical metabolic pathways in a nutshell. So that's what this cycle is all about. The same thing is the reversal as well, you have to think about it, okay. Some of them are feeding to TCA cycle, some of the products are used these intermediate to synthesize other compound, okay. Now, this is some of the condition where the low ATP, how this pathway is being activated, 1, 2, 3, 4, which I explain, and these are the different chapters we are going to see. Next chapter, oxidative phosphorylation, we are going to study next week, okay. And by the way, there is no exam, which I mentioned the syllabus, no exam next week. The following week we will have it, okay. I mean, when is the exam according to the syllabus? Next week? Remember? We are postponing because we missed one class, so I put a revised syllabus for you, for the schedule. So we will 1 week postponing for everything, the exam and everything. Granger collection is again the 4, right? The sale of 1996. Yeah. Do you know what is the story on this one? No? Okay, dihydrolipamide and arsenite. Again, arsenite is also regulating the pathway, and dimercaptopropanol, otherwise called BAL, that is again the mercurial compound as well. They also, involved in the regulation of this enzyme, okay. It's a main inhibitor. Toxic compound, arsenate chelate on enzyme, regulation, arsenite chelate restored the enzyme. Citrate synthase, aconitase, the same thing, how the pathway, again, the reactions, TPA and melanate, oxaloacetate, the compound, and weeks of isocitrate lyase and gene direct. These are some of the experiments they did with wild type and other mutant form. Okay. Now, the next one, which I would like to get into the clinical situation, the regulation of the TCA cycle. TCA cycle is regulated by PDH enzyme, which I mentioned earlier. This regulation by allosteric, allosteric. What is allosteric enzyme reaction? Allosterically, it be inhibitor or regulator, meaning a reversible of phosphorylation. Suppose if there is an enzyme like this, it will go like this, okay. This is the active site. Active site. So the substrate will bind here and then, you know, it bind here, substrate. This is the active site where the reaction is going on. Normally it is okay, okay. Suppose if there is an inhibition, sometimes like the substrate, another molecule can bind instead of the original substrate. That is called competitive inhibition of this enzyme. The another kind is it may bind somewhere else other than the active site and thereby inhibit this reaction. That is called allosteric. Allosteric enzyme inhibition, or this enzyme inhibition is allosterically controlled. Okay. So another way of controlling the PDH or the pyruvate dehydrogenase enzyme complex is allosteric inhibition or allosteric regulation. The defects in the TCA cycle and combines to form the development of cancer. Defect in your biomedical sciences course, defects, okay, of TCA cycle. This is very important for your course. Defects of TCA cycles, meaning some enzyme deficiencies and everything. For example, SDH, succinate dehydrogenase enzyme, fumarase enzyme, and pyruvate dehydrogenase enzyme and kinases. Okay, they form, if any mutation on this enzyme, that form cancer. That's very simple. If there is a mutation in succinate dehydrogenase, mutation in fumarase, mutation in pyruvate dehydrogenase complex, and mutation in kinase reaction, yes, there is a development of cancer. So how to stop that, the mutation, or how to overcome that? That's what the cancer therapeutics, different type of therapeutics they are working on. Okay, and defects in these enzymes also enhances the glycolysis. Okay, defects on these enzymes, okay, are defects of the glycolysis. Promote the glycolysis. Meaning in the cancer cells, it increase of glucose catabolism, glucose breakdown, I put it over, breakdown. Why? The cancer, it really takes much of the energy for the synthesis of small daughter cells and uncontrolled growth. That again another piece of information. When it will do it, it will also produce what? When you have uncontrolled cell growth and more of oxygen is used. Suppose I'll tell you one example. Suppose this is the cell. Okay, the blue is the normal cell, and how many cells are there here? Now we can count it on here, like 1, 2, 3, 4, 5, 6, 7, 8, 9 cells on this particular area. Okay, so now I will give you another scenario, the same area, but what happened here? So if you imagine the TCA cycle on glycolysis and much of the cells or the function and the consumption of oxygen for this particular cells. See, suppose if this is the oxygen supply, the area is the same, and here also the area is the same. So what will happen? Imagine here the amount of oxygen is generous. It can easily breathe. The cells can take up all the oxygen, breathe easy here in this one, and it will produce normal of ATP. But here, what will happen? Less amount of oxygen, but more amount of the cells. What will happen here? The stress. And at this particular stress situation, it will induce these cells, the middle of the cells, you will have hypoxic. Meaning the oxygen for these cells will not sufficient. Those oxygen is coming up, but it is not sufficient. What will happen? These cells, they learn the trick, and they are very crooked cells. These are the cancer cells, and they will produce the hypoxic inducive factors, HIF. Okay. Now that HIF will in turn increases vascular, smooth muscle cell proliferation. Vasculoendothelial, VEGF1. Vascular endothelial growth factors. It will produce more of this, and this vascular, it will promote more of capillary. Capillaries are the blood vessel formation. In other words, the blood vessel, the blood supply to the cancer tissues over here. So over here, and then it will produce more of the blood supply here. And thereby, these cells will get excess of oxygen for this to produce this uncontrolled growth, and this is going to be very growth. So it will grow fast. So the HIF is very important, and that is also one of the factors derived from the TCA cycle of the products it will give you. Okay. The pathway of integration in your textbook, in your figure, and you also get in the thiamine, in the vitamin B1 deficiency, you get beriberi disease. So these are the beriberi disease is one of the deficiency, nutritional vitamin deficiency B1. You get beriberi. This is another biomedical importance for your class. Okay. Also the mercury and the arsenic poisoning. Okay. Also it looks like a beriberi. And thiamine deficiency lead to TPP less. Thiamine pyrophosphate less. Meaning TPP is the prosthetic group for your pyruvate dehydrogenase enzyme complex, PDH, correct? And they are present, the thiamine mainly present in the unpolished rice. I'll write it separately so that you know. Suppose this is the rice grain, and the middle part is the grain, and it's having its own the cover with some the brownish cover. That's why the brown rice is good, right? So because the brown, that's the thiamine. Thiamine. Meaning if you remove the hard shell and then unpolished rice is good. That means brown rice is very good because you have more of thiamine. In some place of nutritional deficiency, you suffer from the beriberi. And sometimes the alcoholics, if you take excess of alcoholics, they also suffer from thiamine deficiency. So what you have to take, if you excess of alcohol, so they have to take more of thiamine as well, vitamin B1. Okay. And they are also, beriberi is a neurological disease. So we don't want to get into that trouble, neurological diseases. And in some of the plants and animals, you have got the glyoxylate cycle. That means this is dicarboxylic acid cycle, not tricarboxylic acid cycle. That's what TCA cycle, in other words, called tricarboxylic acid. This is called dicarboxylic acid cycle. Dicarboxylic acid cycle. They are present in bacteria. These are present in bacteria and some plants. You will see later on the dicarboxylic acid cycle. Okay, and the most of the class, which what we have done so far, the TCA cycle plays a major role in the intermediary metabolism. Okay, for this particular class, okay, as I mentioned earlier, that your homework is going to be another first 20 questions from your back of the textbook. Okay, so that's what your homework assignment. And I will give you a bonus question for this particular class is the drug design. Okay, a drug design. I will give you an article, a review article, and there I have given glucose plus citrate. It prevent the NSAID-induced ulcer. I'll write it separately. I'll write it. NSAID, non-steroidal anti-inflammatory drugs, because you are biomedical science students, right? Non-steroidal anti-inflammatory drugs, it produces intestinal ulcer, okay? Intestinal ulcer, if you take chronic one, okay? But if you add citrate and glucose plus this NSAID, plus NSAID, and what happens if you add together and no ulcer? Okay. So the mechanism lies within the glycolytic pathway and TCA cycle together. Okay. It is my own research paper which I did in England, and I will show you now. Where is it? Okay. Where you will find it. If you go on to the course content, okay, go on to electronic reserve, and here you have a password. You copy the password, copy, okay, click on the electronic reserve, and you will find out this link, and you just scroll down and you will find a 1995 article, mitochondrial damage. That's we will discuss next week on that part. And this one, biochemical basis of non-steroidal inflammatory drug-induced damage to gastrointestinal tract, a review on the hypothesis, Scandinavian Journal of Gastroenterology, okay? And this one, let's see, open. Coming up now. So I have to paste the password here and click OK. Yes, now I get the paper now. And this article is a challenge. When I landed in 90s, and then 2 to 3 years I was working on to break my head. And luckily I saved myself. And getting on the mechanism, how on earth the inflammatory drug, anti-inflammatory drug produce an inflammation. It is an anti-inflammatory. It should not supposed to produce any inflammation, but it does. So that's the question. And then how it is being changed. So if you read this review, okay, I need a comment read the hypothesis, okay, and you will get some questions on this, and this question for your bonus one. You just read now and answer. I'll show you the review. Just read and understand. This is the mechanism of the hypothesis, how the inflammation is produced with the NSAIDs, and the last one I'll show you. This is again from mitochondria, okay, mitochondrial uncoupling of oxidative phosphorylation, and this is the nice phase of this class. And the next class, or next week class, a bridge. So if you study this, it will help you to understand the next part as well. Okay, this is more of applied one. Whatever you studied in your theory lecture in a textbook, and this is on a research paper. Then you can see here the glycolytic enzyme here in rat small intestine, which I measured it. Glucose 6-phosphate dehydrogenase, phosphoglucomutase dehydrogenase, aspartate carbonyl transferase, thymidine, phosphorylase, hexokinase, peroxidases, all this one. If you see the read the text, you will find out all those details. And the mechanism. And here, this is the one which the neat part. Because the physician, and then he treated the patients with the glucose and citrate with the indomethacin, that's one other anti-inflammatory drug. He did not find any abnormalities. He did not find any ulcer on it. But at the same time, if you give only indomethacin, for the only NSAID, the patient become bleeding from the intestine. But the American Journal of Gastroenterology did not accept the results because there is no logic behind it. So he gave that paper to me, work out the mechanism, because I was really a biochemist at the time.
when I was not teaching biology, but I was the clinical biochemistry in a medical school. So I was, working on all these 2 years on this particular problem, and finally I found out the phosphofructokinase PFK that inhibitors over here, citrate and ATP. And then if you read the paper, you will find the glucose will feed to this glucose 6-phosphate, and then phosphofructokinase inhibits the reaction, and this will be shuttled to glucose 6-phosphate dehydrogenase to HMP pathway, that hexose monophosphate shunt. So this will produce more of a NADH and NADPH reaction, and increasing the potential, and the mitochondrial level will be altered, and you will become a unhealthy cell to the healthy cell. So then they review or accepted this hypothesis, and that's why we can publish this work. And it works. It gives all the experimental evidences that's true. So how the glycolysis and the TCA cycle are linked together, please go through this particular paper. Mainly this one is involved in the citrate, and then as well as in a glycolytic pathway. Okay. I need 1-page report with concentrating on the hypothesis and this pathway. Read this one, and for the exam you may get some questions from this pathway which is more relevant for this class. Okay, and also it will give some additional information of inflammation and how to control the inflammation. These are my own work we did earlier. So you know how to get this article, and you know how to study, and you can also study the first 20 questions on this one and write the summary of this particular research paper. Any questions? If you have any questions, Alejandro, Mary, Komal, Nguyen?
Dr. Soma.
Yes.